We offer a robust platform to produce blood cells from human pluripotent stem cells.
The main advantage of this technique is that we get consistent and precise yields of hemogenic endothelial cells.
We expect that our platform will enable broad applications in disease modeling and screening for therapeutic compounds. This technique is particularly beneficial for hematology and immunology research. The method can be applied to genetic intervention or gene editing of human blood cells. It allows for easy induction of vectors in hemogenic endothelial cells.
The most crucial point is LM511E8 coating for tiling PSC spheroids. We do not recommend skipping the coating or substituting with another matrix. Visual demonstration is important because we want to show how to rinse the cells with a dissociation buffer and how to coat plates with LM511E8.
Grow human pluripotent stem cells, or HPSCs, to between 70 and 80% confluency on an LM511E8-coated 6-well plate in MTs or 1 medium. 4 days prior to hemogenic induction, aspirate the medium and wash twice with PBS. Rinse the cells with dissociation solution and incubate at 37 degrees Celsius for 15 minutes. Resuspend the cells in PSC maintenance medium and transfer the suspension to a 1.5 milliliter microcentrifuge tube. Centrifuge the cells at 200 times G for 3 minutes. Aspirate the supernatant